Hello friends, this video on Principles of Inheritance Part 14 is brought to you by examfear.com. No more fear from exam. What is codominance? Now you might be thinking then that again how come there is monodominance? Now as I said, Mendel gave his laws based on the experiments which he performed on the pea plant. But after Mendel, a lot of scientists performed a lot of other experiments, and that is how they gradually got to know that what all additional facts were still there. So once it came up with the concept of incomplete dominance, then it came up with another concept of codominance. Now co means something, coordination, that means something which happens together, right? So anything that goes together, that is called, that has this co in before the word. So codominance is also when both the parents dominate together. Now in incomplete dominance we saw that none of them could actually dominate, so they blended to give an intermediate phenotype. Here we will see that both the parents will dominate and both the parents will dominate together. So let us see what is codominance. So it is occurrence of a mixed phenotype. So please understand the difference between intermediate phenotype and mixed phenotype. Mixed means you will have the dominant trait will also get expressed, the recessive trait will also get expressed. So both the traits will get expressed, both the parents will get expressed. So that is mixed phenotype. In a heterozygous obtained when two homozygous parents of different phenotypes are crossed. So something like this. Here you can see a red flower. If you cross it with a yellow flower, so one option is, as per Mendel, you can get red flower because red is dominant over yellow. So that is as per Mendel. As per incomplete dominance, you neither get red, you neither get yellow, you get something like orange, something in between. So that is incomplete dominance. But if you get something where half of the petals are red and half of the petals are yellow, what does that mean? That means that both of them are being expressed. Even the red is being expressed, the yellow is being expressed. So this is a mixed phenotype and this is called codominance. That means both the traits are dominating and they are equally dominating. So here F1 generation resembles both the parents. So you cannot say that this is an intermediate because it has some traits which exactly matches with this one. It has some features exactly matching with this. It has some features exactly matching with this. So it resembles, the F1 generation resembles both the parents. Here both the alleles are equally expressed. As I said, no one is dominating over the other one. Both are getting equally expressed. So let us take an example of codominance, and the human ABO blood grouping is the best example where we can see codominance. So let us look at what happens in the blood grouping. Now ABO blood groups are controlled by a gene I. So the gene which controls your blood group, that means this is the gene which actually decides which blood group you are going to have, right? So this is the gene I. Now this gene has 3 alleles, like how you saw in case of the pea plant. You saw that everything, most of the things had 2 alleles. Whether you talk about the seed color, whether you talk about the seed shape, you talk about the height of the plant, everything had 2 alleles. But here you see they have more than 2 alleles. The gene has 3 alleles, and the 3 alleles are IA, IB and I. So these are the 3 alleles. Now what is the difference between these 3 alleles? Now all of them will be used, or they actually control the synthesis of proteins. Now IA produce sugar, IB also produce sugar, but it is slightly different from that produced by IA, whereas I doesn't produce any sugar at all. So that is the actual purpose of each of these alleles. So that is how their functions are different. Okay, now what are the various genotypes possible with these alleles? Now each person, each individual will have any 2 of these 3 alleles. Everybody will have, because we all know, right, a gene is made up of an allelic pair. So 2 alleles make a gene. So one person can have any 2 alleles. They can either have IAIB, they can have IAI, they can have IBI. So any 2 out of the 3 alleles can be present in an individual. There are various possibilities, various combinations can be had with these 3 alleles. So let us have a look at that. Now, what are the various possibilities? Now, let us suppose these are the 2 parents. So now, if one parent if the genotype of the offspring, so this is the offspring I am talking about, right? So if we say that the offspring has a genotype IA, so what does that mean? What will the parent have? Again, there are possibilities. The parent can have IA. So that is one possibility that the parent can have. If the genotype is IAI, that means the parent has IA, the parent too has I. If the genotype is IAIB, that means parent 1 has IA, parent 2 has IB. Similarly, in this case, we can find out all the possible options for the two parents, because from there only the genotype is coming out here. So maybe both of them have I here. So here again, if you see, maybe this has I and this has IA. Similarly, here maybe I and this is IB. So these are the various possible options that can happen with the parents. I mean, depending upon the blood groups of the parents, the blood group of the offspring will be decided. Now the question is, now as you see here, in some cases, both the parents contribute the same allele. So in that case, we know what will be the blood group. So if the genotype is going to be IA, the blood group is going to be A. Similarly, if it is going to be IB, the blood group is going to be B. Now what will happen when the alleles are different? So in the heterozygote scenarios, what is going to happen there? So for that, we should have knowledge on the dominance of the various blood group alleles. So what are the possible? I mean, who will dominate whom? So let us talk about dominance. So IA and IB are completely dominant over I. Now why are we using the term completely dominant? Because now we have two concepts. One is complete dominance, the other is incomplete dominance. So IA and IB. So if you have a scenario where I is present, that means it is going to have blood group A. Similarly, if the genotype is IBI, that means it is going to have blood group B. So I is going to be the recessive allele. Correct? So IA and IB will be completely dominant over I. Now what about IA and IB? What if a person has a genotype IAIB? So who will dominate? IA will dominate or IB? So IA and IB, that is the example of codominance. They are codominant with each other. So IA will also get expressed, IB will also get expressed. And as a result, what would be the blood group? The blood group would be AB. And that is why we have 4 different types of blood groups, right? There are 4 blood groups which are seen in human beings. And what are they? A, B, AB and O. So when do you get A? When the person has IA. So when the IA allele is present. Similarly, blood group B, when IB allele is present. This when both IA and IB alleles are present. And O when I is present. So O is the scenario where no sugar is being produced because only I is present. Right? And AB is when IA and IB both are present. B is when IB is present and A is when IA is present. So that is how we get the 4 different blood groups. Now let us make use of the same table and let us try to see what would be the blood group for each of these genotypes. So the blood group is nothing but the phenotypes. That is what we actually see. The one that is that trait which gets displayed to us is phenotype. So here blood group is going to be our phenotype. So if the genotype is IAIA, so homozygous both has IAIA, so blood group will be A. If it is IAI, as I said, IA is completely dominant over I, so it is again going to be blood group A. If it is IAIB, they are co-dominant, so both will get express themselves. So both will equally dominate, so it is going to be AB. IB again it will be B. Similarly, IBI again is B. IAIB again will be AB. II, this is going to be blood group O because it doesn't have either of the sugar producing alleles, IA or IB. IAI is again going to be A and this is again going to be B. So this is how the blood group is determined, that which individual will have which blood group. And also the parents play a role because at the end of the day these alleles are coming from the parents. So one allele is coming from the mother, the other allele is coming from the father. So you will actually get to see a lot of questions coming out of these blood group and this blood group scenarios. So where you will be given the alleles of the parents, you will be given the blood group of the parents, of both the parents, the mother and the father, and then you will have to say whether this person with a specific blood group can be a kid of those parents. So there again you will have to find out what are the possible offsprings or what are the possible blood groups which are possible with those alleles of the parents. So this is how the dominance in blood group is studied. So here if you look at the genotypes and the phenotypes, what do you see? If you look at the phenotypes, how many unique phenotypes are you able to see? So if you talk about phenotypes, there are 4 unique phenotypes. And what are those 4 unique phenotypes? A, B, and O. And these are the 4 blood groups which are seen in human beings. So that's about phenotypes. Similarly, if you talk about genotypes, so in genotypes, how many unique combinations do you see? So here if you see IA is one unique combination, IAI is one unique combination, IAIB another unique combination, this is another one. But from this, if you see, this is a repetition, it was already there. II is a unique combination, this and this are again repetition. So total you have 1, 2, 3, 4, 5 and 6. So 6 unique combination of genotypes. Right, so you have, with the 3 alleles of the blood group gene, that is IA, IB and I, you can have 4 unique phenotypes and 6 unique genotypes. And what are the 6 unique genotypes? This is IA, IB, IB, IAIB, IAI, IBI and II. So these are the unique genotypes. Thank you. Please visit www.examfear.com to watch more videos, attempt free online tests, get free study material, find tutors and mentors. Thank you once again.